Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I have with me Melissa Sherman, CEO of MOBILion Systems, a company pioneering next-generation separation science by innovating best-in-class instruments that deeply, accurately, and efficiently characterise complex molecules. Welcome, Melissa.
Thank you. It's my pleasure to be here today.
Hi, this is Chris O'Brien. We'll be right back with the rest of this episode in a moment. I wanted to introduce you to the BioRossi Few and Far Between podcast, hosted by me. Few and Far Between brings you conversations from the front lines of drug development. Each episode features cutting-edge discussions with top biotech innovators and clinical trial industry experts. You can find our episodes on Apple Podcasts, Spotify, and other streaming platforms. Thanks for listening, and now back to the episode.
So today we'll be discussing the growing interest in next-generation sequencing, or NGS, and mass spectrometry. But firstly, Melissa, perhaps you could tell listeners a little about yourself, the passions and interests, as well as the opportunities that led you to do what you do today with MOBILion.
Sure. Well, I'm a polymer chemist by training. So I had my PhD in polymer chemistry and was always interested in science and how things work, but probably more interested in the practical application of science. So taking science, productizing, and commercializing it to build businesses. I've been an executive for the past 25 years in companies ranging from large global public companies to emerging growth private companies, and now with MOBILion, a venture-backed startup company. So my passion is building and growing businesses, and I was very much attracted to MOBILion because I think it's somewhat of a once-in-a-lifetime opportunity as a scientist to be introduced to a platform technology that has the opportunity to be so disruptive in the industry and to have the privilege to be able to productize and commercialize something that is that seismically shifting, I guess, I really think is a once-in-a-lifetime opportunity. So I worked for the early stage technology investment company that made the original investment in MOBILion. And when it was time to set up the company and take it forward, I left the investment company to be the CEO of MOBILion and take it forward to productize and commercialize the platform technology.
Thank you, Melissa. Yes, as you say, it is something seismic indeed, this technology. So if we sort of go into the history of mass spec for a second, it was originally employed in atomic physics, and it's now an indispensable tool in modern science and, importantly, medical science. Indeed, according to the biomedical scientists, the genesis of mass spectrometry can be traced back to the late 1800s and the discovery by Joseph John Thomson of the electron and measurement of its mass, from which the basic technique to measure the mass-to-charge ratio of ions was developed during the 1920s, and the history of mass spec has since been driven by numerous pioneers, such as Francis Aston, Alfred Neer, and Wolfgang Paul. But Melissa, tell us about MOBILion's work with mass spectrometry technology at the moment.
Well, I think it's important to note that more often than not, at least in healthcare and for complex sample analysis, that there's a separations technology integrated with mass spectrometry. So the gold standard instrumentation that we talk about and the industry that we're most disrupting is the liquid chromatography mass spectrometry industry. And the liquid chromatography is the separations piece that you separate complex samples before feeding them into the mass spec, which serves as the detector. And so, liquid chromatography has been around for 35 years. If you think about it in the way that it works, it separates liquid samples, so liquid phase kinetics, which are slower than gas phase kinetics. And it's a lot of pumps, tubing, solvents, separating analytes with chemical properties, hydrophilic, hydrophobic, and other chemical properties. And so our separations technology is based on an invention at a United States federal laboratory. It was invented by Richard Smith at Pacific Northwest National Lab, and it is a new category of separations called ion mobility. separations, which separate in the gas phase. So gas phase kinetics versus liquid phase kinetics means it can go much faster, 1,000 times faster. And the other aspect of it is analyte agnostic and separates based on inherent physical properties of molecules. So an inherent physical property of a molecule is more reproducible, more accurate. And so it's an entirely new way to do separations, really addressing the speed, specificity, and complexity constraints of the incumbent of liquid chromatography. So we say generally with this separation science and separation technology that MOBILion is advancing, that we make LC mass spec workflows better and we reveal what it leaves unseen. So we provide faster, more accurate characterization, and it's applicable across all areas where LC mass spec is used, which does include healthcare, life science, drug development, discovery, biomarker discovery, but also is applicable in the food and environmental testing markets as well.
Thank you. Gosh, a lot to take in there, and thank you for breaking down the more scientific aspects for those of our listeners who perhaps aren't familiar with the technology. So if we go broader, what is the practical impact of this new tech for researchers and drug development specifically?
Yeah, well, I can give you some examples. So let's just start with the impact on drug development. So if you, and this is part of the regular drug discovery and development process, right? So if a researcher can better understand and more accurately characterize the patient's target biology and the chemistry of the drug that they're developing, it results in safer, more efficacious drugs being developed. So, right, it's all about understanding what is the biology of the patient and of the disease that you're trying to treat. And you need to understand what that biology is and what that mechanism is. And obviously, if you have a disease, it's a normal, healthy mechanism that goes sideways and turns into an unhealthy mechanism. And it's critical to understand that biochemistry and that biology. And so these tools, analytical instruments, are used. And again, LC mass spec is the gold standard instrument when you're looking at proteins, peptides, lipids, glycans, and metabolites. So we're not talking about genomics. We're talking about everything downstream from genomics, which is just as critical, if not arguably more so. So you have to understand the patient biology, and you have to understand the biochemistry that changes when you go from a healthy to a disease state. And when you understand that, then that becomes your drug target. Once you identify that target, then a whole slew of medicinal chemists are set out in pharmaceutical companies to develop drug chemistry that affects that target. And so the more accurately you do that, right, you can understand that if you are doing high throughput screening of a number of potential targets and a number of potential drug candidates, the goal is to have the most accurate target and the most accurate drug candidate to affect that target in a positive way and stop that disease process. And so the more accurately you characterize that patient biology and the more accurately you characterize those drug candidates and the faster you do it, it saves time and money and results in more accurate drugs, less failures downstream. So when you think about having instrumentation that kind of breaks the barriers of incumbent characterization, right? You can think about making these go, no-go decisions within a drug development program at earlier stages and prior to when significant resources have already been invested is critical. And then the accuracy of, like I said, validating that target biology and the drug chemistry is critical to having the best target ID and the best compound that you move forward into that program. So doing it faster and with fewer iterations saves money. If it's more accurate, it has the potential to reduce, say, the number of cell studies, to reduce the number of animal studies, and increase the probability of success when it goes into the clinic. So when you think about a drug development program, right, on average, a drug development program before it even gets into human clinical trials costs an average of $1 billion and takes 4 years just to get it to be accepted for clinical trial phase. And 85% of those candidates and 85% of those drugs moving forward fail. So that's a spend of what? If you take $1 billion in 4 years, it's $20 million a month. If you can shave a month, right, you're saving $20 million. If you can increase the accuracy and depth of that characterization, you can potentially minimize that high failure rate when you submit drugs further down the drug development process, which becomes even more expensive. I can give you some examples of specific examples in the drug industry where our MOBI product was used to help do exactly what I've been describing. So one example, and these are things that our customers and partners have talked about. So Genentech, for example, they make an antibody drug conjugate, which is a protein that has a linker, and they use the linker to tie onto a cytotoxic cancer therapy. And so the reason they use this antibody drug conjugate to deliver this drug is that it gets the drug delivered specific to the target and not systemically throughout the patient. So when you have a therapeutic with a lot of side effects, you don't want it to be systemically delivered to the body. You'd rather just have it be delivered to where the target is, to that diseased cell. And so Genentech had this antibody drug conjugate linker molecule, and the program was stalled because they knew that they were not adequately characterizing it. They knew this linker had two forms. One was active, one was inactive, but they could not separate and identify these two forms. They knew it was there from the theoretical chemistry, but they couldn't see it with the analytical instruments, and we were able to see it and separate and identify those two forms. So then Genentech was able to move this drug program forward, and they could establish the right purity, dosage, and manufacturing process to synthesize that linker as a drug delivery vehicle. Another example, this just happens to be at Genentech as well. So they were developing a small molecule drug as a KRAS inhibitor. So this is a cancer therapeutic that was being developed to inhibit a specific mutation on the KRAS oncogene. They struggled to characterize it because, again, it had two different isomeric forms. So basically this drug has two forms that has the exact same mass, but it's a conformational isomer, which is very difficult to separate with liquid chromatography because it's the same chemistry. And then it can't be separated by mass. So Genentech needed to add another separation technique that is a different way to do separations to be able to separate these two small molecule drugs from one another. And again, one of the forms of the drug is effective, the other form of the drug is ineffective. And they were able to use the MOBI platform to be able to separate those two forms of the drug and be able to adequately characterize it and ultimately able to manufacture it and monitor the production of it as well. That happens to be Genentech. A couple other examples in the peptide therapeutic space. So peptide therapeutics have become very popular recently with the GLP-1s. These kind of magic weight loss drugs that started as diabetic therapeutics. But there are a number of strategies used in peptide therapeutic drug engineering and formulation. So you can take linear peptides and make them cyclic to change the stability in the body. There's other engineering techniques where you can add D amino acids instead of L amino acids to improve, again, in vivo stability and create secondary structure and tertiary structure that mimics proteins. So there are a number of different strategies that are emerging in peptide therapeutics to make them more effective, make them more stable, ultimately even try to get them to be suitable for oral dosage instead of injectables. And so what that adds is just a level of complexity and characterization that today's instruments can't achieve adequate separation and identification. In some cases, they can achieve it, but it lacks the specificity, or it requires really long analysis times and really expensive analyses. So as an example, Merck, who was working with a peptide drug formulation, used our platform instead of LC mass spec to eliminate costly and lengthy sample prep steps. So because we have a different way of separating, we were able to eliminate costly and time-consuming labeling that's required for liquid chromatography separation, and it saved them 8 hours of analysis time per 86 well plate, in addition to giving more accurate results and more specific results from a characterization perspective.
Thank you, Melissa. So just to be clear, it is this specificity of separation that is setting apart MOBILion's technology. So you sort of referenced some big names there, but I'm also aware that this technology is so too being used in an academic laboratory setting as well, isn't it?
Absolutely. Yes. We have academic researchers as well as pharmaceutical researchers, as well as food testing companies, as well as environmental testing companies and agencies. So it is really a next generation separation science. And like I said, when you look at who uses liquid chromatography, mass spectrometry as the gold standard, academic researchers, government researchers, pharma company researchers, agencies like the FDA and EPA, food testing companies, environmental testing companies. So it's used by a lot of scientists to characterize molecules, again, downstream from DNA, downstream from genomics. It really is the gold standard platform. And so, like I said, we make that workflow better. So inserting our technology in that workflow gives extra information, faster information, more reproducible, more specific, to just take the characterization of whatever the researcher is doing and make it better, faster, more specific, more accurate, and more reproducible.
Thank you. And so with this potential now, what of the future? In your opinion, what next for NGS?
Yeah, so, I really think that it's the complement, right? I mean, everyone talks about next generation sequencing, and it definitely is step one of human biology, right? DNA makes RNA, which makes proteins, and proteins control cellular function, and cells are made up of lipids. And, when you look at healthy cell processes versus disease cell processes, you're looking at all kinds of things. You're looking at glycans, you're looking at lipids, you're looking at metabolites. And so you can't really look at one aspect of that human biology and get all of the answers. And so the more tools we have to better characterize the holistic human biology, the more discoveries will be made, whether it's better drugs, whether it's better biomarkers, right? So biomarkers are important signatures of health, disease, and drug response. So it's really important that we understand that entire paradigm of human biology. So I don't think one is better than the other. I think they're complementary. I think what we have to appreciate is that, looking at genomics alone is a very incomplete picture. And so it's step one of that human biology process. But there's so much more that we need to understand to truly impact how we predict, diagnose, and treat disease. And genomics is just one piece of it. I mean, if you look at genomics accounts for maybe 20% of disease, so what about the other 80%? You have to be looking at these other molecules downstream from DNA and RNA to truly impact how we predict, diagnose, and treat disease. And that's what we're enabling.
Thank you, Melissa. It's been a pleasure.
Thank you.
And that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on Apple Podcasts, Spotify, Overcast, Pocket Casts, Podbean, and pretty much wherever else you download your other podcasts from. Just search for pharmaphorum and follow and hit subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us on Blue Sky and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.